Long before we built computers out of silicon and copper, nature programmed the ultimate operating system. It's running right now in every living cell. You spend your days optimizing algorithms and managing silicon-based datasets, but biology offers a far older and more complex architecture to explore. The challenge for the next generation of engineers is learning to interpret and interact with this organic source code. Our first attempts at patching this biological software have arrived as modern vaccines, highly targeted medicines, and rapid diagnostics. We are beginning to use technology to debug failing biological systems. To understand the system, we must first extract the data. Diagnostic tools like PCR or polymerase chain reaction act as a microscopic search and copy function. By finding a specific sequence of DNA and amplifying it millions of times, we turn invisible genetic fragments into readable datasets. Much like early computing, this biological hardware started out bulky and rigid. This early PCR machine from the 80s, with its primitive screen and chunky keypad, represents the moment we began digitizing the genome. Mastering these diagnostic tools transformed biology from a purely descriptive field into an analytical one, providing the high-resolution data necessary to eventually treat life as a predictable, manageable system. We have moved past simply reading genetic code. In the modern era, the objective is to actively edit it. Tools like CRISPR now function as a precision text editor for the genome. Scientists can physically locate genetic defects, snip them out, and replace them with corrected sequences to prevent inherited disease. This shift has triggered the expansion of a massive industrial ecosystem. What was once confined to a few academic labs now spans a global network of clinical research, regulatory affairs, and specialized R&D centers. This growth creates a critical need for computational minds. The biotechnology sector now generates more genomic data than biologists can manually process, requiring machine learning to find the patterns hidden in billions of base pairs. The industry has evolved into a multidisciplinary tech landscape, where biological insights are only as powerful as the computational models that support them. Treating life as editable code grants us immense power, but it also creates significant societal friction. Every genetic edit raises difficult questions about where we should draw the line between therapy and enhancement. These ethical concerns are most visible in our food supply. By genetically modifying crops to resist pests or drought, we've improved yields, but we've also sparked deep public skepticism about the safety of our dinner plates. This public fear is visceral. Protests against frankenfoods illustrate the resistance that occurs when genetic engineering reaches consumer products without clear societal consensus. There are genuine environmental risks to consider. Cross-pollination between modified crops and wild species could introduce engineered traits into the wild, potentially threatening the biodiversity of local ecosystems. To manage these risks, the industry relies on biosafety, a strict regulatory framework of containment protocols designed to prevent accidental ecological contamination. Without these ethical frameworks and biosafety standards, biological innovation could easily outpace our ability to control it. They serve as the necessary cybersecurity for our natural environment. The global COVID-19 pandemic served as a high-stakes stress test for this entire system, forcing the industry to move from genetic discovery to mass production in record time. The physical result of this global effort was the deployment of mRNA vaccines, a breakthrough that relied as much on sequencing data as it did on traditional chemistry. mRNA technology delivers a sequence of genetic instructions that teach the immune system to recognize a specific viral protein. It allows the body to build its own defense without ever being exposed to the actual virus. This convergence of silicon microchips and biological cells defines the landscape you will soon enter. The tools you are learning today are becoming the primary instruments of biological discovery. We see this in the way AI now predicts three-dimensional protein structures. By modeling complex folding patterns in seconds, computational tools help researchers design targeted molecular therapies that were previously impossible to visualize. As the line between hardware and wetware continues to blur, the engineering of the future will be as much about organic cells as transistors. Success in this field requires moving beyond syntax and logic. To lead in this new era, you must master the ethical and environmental frameworks of the living systems your code will soon influence.